Hello, I am Dhinakar Kompala of Sudhin Biopharma Company in the center, with Christopher Glascock on your right and Braden Smith on your left. We present a short 15-minute video on a novel method we have developed for clarification of cell culture broth using Sudhin BioSettlers. We begin with a series of 4 photos taken during a continuous or flow-through clarification of high cell density culture broth from a Fed-batch bioreactor at a big biopharma company's process development lab. In this first photo, you can see a big square reservoir bottle in the center, initially filled with about 15 liters of acidified culture broth before the liquid gets pumped into the BioSettler 150 shown on the left. You can see a strong gradient of CHO cell clumps inside the BioSettler during the flow-through clarification process, with the more concentrated cells accumulated at the bottom, while the top of the BioSettler is filled with a highly clarified supernatant. Pumping culture broth into the settler from its bottom side port gently pushes out the clarified supernatant through the settler's top central port. In this second photo, a bioprocess engineer is examining a collected bottle of clarified supernatant to see if there are any CHO cell clumps at the bottom, while the reservoir is being tilted to pump out the culture broth completely. Of course, there are no clumps visible at the bottom of this bottle. In the third photo shown on the right panel, you can see the clarified supernatant remaining in the BioSettler after pumping in all the 15 liters of culture broth. This remaining supernatant is being aspirated with a tube inserted from the top side port. Soon after the inlet flow of culture broth has stopped, we can see a loose pellet of settled cell clumps packing into the bottom of the BioSettler. In the fourth photo shown on the right panel, you can see most of the remaining clarified supernatant has been harvested by gently lowering the aspirated tube from the top side port. At the end of this 4-hour flow-through clarification of 15 liters of culture broth, the packed cell pellet at the bottom of the BioSettler is very well defined, as if the settler has been centrifuged at high G force. Instead, our BioSettler operates at 1G, the Earth's gravitational force, and demonstrates an enhanced settling soon after the inlet flow has stopped. Preliminary data collected during my visit showed that the turbidity values for each of the collected supernatant samples were low, around 100 NTU, while the starting culture broth had a much higher turbidity of over 2000 NTU initially, indicating a large reduction of over 95% in turbidity. This significantly clarified supernatant can be processed more easily during a final or secondary clarification step using a depth filter. In the next slide, my colleague Chris Glascock will present a more complete data from one of our own clarification experiments conducted at Sudhin Biopharma's lab in Colorado.
Hello, I'm Chris Glascock, a research scientist here at Sudhin Biopharma. In this slide, we present more extensive data from a clarification experiment conducted at our own lab about 2 years ago with a relatively lower cell density culture broth. Starting with about 9 liters of cell culture fluid, having a turbidity of only 528 NTU, we pumped in pre-acidified culture broth and collected the clarified supernatant in 6 fractions into 1 liter bottles and analyzed the antibody and total protein concentrations, as well as the turbidity and packed cell volume for each liter sample. After pumping out all of the culture broth, we were able to obtain about 2.8 liters of clarified supernatant remaining in the 3.8 liter volume BioSettler 150 and we also analyzed its contents. In summary, we have recovered over 95% of the secreted antibody in the collected supernatant and about the same amount of total protein. Furthermore, we have observed that the PCV is reduced to 4% of its starting value, and the turbidity of the supernatant has been reduced to about 100 NTU, which is similar to the values obtained at the same flow rates in the previous experiment just discussed. This and an earlier evaluator expressed a desire to test the novel technology in a smaller biosettler using the cell culture broth from their 2-liter bioreactors in their process development labs. In the next slide, my colleague Braden Smith will discuss these new designs.
Hello, this is Braden Smith, Director of Device Design and Production here at Sudhin BioPharma. Our BioSettler is now offered in a smaller format, ideal for process development. Our new design uses a 75 millimeter extrusion in various lengths, providing several volume options. These range from 250 milliliters up to 2 liters. Future designs will scale up to tens or hundreds of liters. Shown here are 500, 1,000, and 1,500 milliliter units. The smaller 500 milliliter unit shown on the left is expanded, showing the internal cones that act as inclined settling surfaces. At the top, there's a set of aspirator tubes used to harvest the remaining supernatant. In this next slide, you can see a close-up view of the aspirator tubes. Each tube descends to a different depth and can be individually selected by releasing its corresponding clamp. In the next slide, Dhinakar will summarize this presentation and add some closing remarks.
With this final slide, we summarize the data presented here and provide summary information on other applications and sizes of our biosettlers now available for purchase, and further additional investigations in your own process development laboratories. Chris Glascock has done numerous clarification experiments using our initial BioSettler 150 at Sudhin Biopharma Laboratory in Colorado and presented a representative data set from one of those experiments in this short video. In summary, we have reproducibly achieved over 95% recovery of secreted antibody product in the clarified supernatant, over 95% reduction in the packed cell volume, and turbidity of around 100 NTU at the flow rates of about 3 to 4 liters per hour that we used to complete the clarification process within a total processing time of under 4 hours. The big biopharma company where we demonstrated clarification successfully and presented mainly the photographic evidence in this short video has subsequently bought a smaller BioSettler 75 polypropylene settler, which we introduced about a year ago. After conducting their next clarification experiment on their own without my presence, they have recently bought the same BioSettler 75, now made out of polycarbonate, so they can see the internal settling processes more clearly during clarification. Here is a quick summary of some other applications for the biosettlers. A cell therapy company has purchased 3 of our initial BioSettler 150s 2 years ago for gentle harvesting of larger organoids, such as beta islet cells. They purchased 2 of our smaller BioSettler 75s earlier this year, and 4 more of the same about 2 months ago. We use internally the BioSettler 75 2000 as a selective cell retention device for a 5-liter CHO perfusion bioreactor to remove dead cells and cell debris selectively, while returning over 95% of the live and productive cells back to the high cell density perfusion bioreactor. We also use a stainless steel BioSettler 300 in our high cell density perfusion bioreactor cultures of recombinant yeast, Pichia pastoris, secreting a heterologous protein product in the supernatant. We have routinely achieved high cell densities in excess of 1200 OD and have operated such high cell density yeast perfusion bioreactors for an extended culture duration, typically over a month or 2. The current industrial practice of using hollow fiber membranes for cell retention has a pain point in either of these two applications, as these membranes get clogged over time, resulting in premature termination of perfusion cultures. A novel application that has been proposed and hopefully will be tested in the near future is to precipitate antibodies to a large enough size, say about 10 microns. So these precipitates will settle faster inside our biosettlers and be harvested continuously in a flow-through process. We can think of several other applications of our biosettler in gene therapy, cell therapy, and biomanufacturing industries. We hope to hear from you by phone or by email, address given here below. If you're interested in collaborating with us on such novel applications, and of course, to use our biosettlers now available in many sizes from 250 milliliters to 20 liters in your own process development laboratories for the now proven application of clarification of cell culture broth from Fed-batch bioreactors. Thank you for your attention.